What we started at EORTC now, let's say a good year ago, is what we call the SPECTA platforms, which are really meant, they're happen in different disease types, and maybe I'll just talk about the colon as one I'm very involved in and as an example, but it's also in brain tumors, melanoma, lung, etc. So the point really is that today you have all these new molecular classifications going on. You know that just no drug is going to work in all patients. This is a given. So you know that when you develop your drugs, either from pharma or academic, you need to take into account all these stratifications. But these stratifications are really difficult to do. They're complex technically. They're probably beyond the remit of a single institution, probably even a country. If you want to offer that to your patients even, it would be really hard to get that organized on a national level. So to bring down all those barriers, the point was, let's just all do it together. Let's put it into one big pot. Yeah? So by centralizing efforts and money, funding, we are able to offer for any country, any tumor that gets diagnosed, all this state-of-the-art profiling and doing it just once. But then everybody can profit from it, in the sense that you have a specific clinical trial with, from one pharmaceutical company wanting to look for patient XYZ, can come in, look for it. Another company looking for another profile on the same patient population can go fish for it. So we're just trying to mutualize this, make it just much more efficient, to decrease the time it takes to otherwise set up these really difficult efforts. If you don't set up these efforts, they just won't happen. Yeah? So these are platforms really meant to mutualize, to increase efficacy. And that's fun, because we get to pour in our science, and then you just see it happen. Yeah? On the science part, we know that there's features that need to be taken into account. Okay, so the fun thing there is that we can dictate kind of which are the features we think are important to measure, and that means we can go very far, very complex, because some of our frustrations otherwise is that even within clinical trials, only very basic features get measured. Then we go like, no, it's not enough. You need to go further than that. So here we can really say, okay, we're just going to measure tons of complicated features. So that input is really fun for us. But the other part then is indeed that this makes it attractive for pharma and academic trials to come in and say, okay, you've given me the resource now with, let's say, 100 alive patients with this very rare feature. I just can go cherry pick them and test my drug in them. And so it's a live platform of annotated patients. And that's the other part where our frustrations are decreased, is the fact that we can run these trials now. Before we had cool ideas, but it seems like an insurmountable mountain to set it up. And yeah, it's just the logistic part is taken care of, that's it. Colon is fun because there's been a lot of changes in the last year. If you remember, for breast and for colon, there have been, and we're talking just about AML here in the session just before today. So there's been substratifications going on for many diseases. Breast, you would never treat them in the same way. So colon, that was really lacking. So part of also our academic work, again in international consortia, was to get a real, biological subclassification going of colon. Intrinsically different biological features that we know those colon tumors, whatever you give them, doesn't matter, from the beginning they're different. Okay, and this, for example, is something we presented at ASCO this year. So it's just very fresh data. It still needs to be published, but you can already put it into your platform, so you know that the next drug trial will take into account those features. These are hundreds of mutations, full gene expression, so very complex features, which again you can do centrally, but you can't just do, you can't expect everybody to set this up in their own lab continuously, or every drug trial again do that. It just would be impossible.
So how do researchers actually access the information?
So it's very open. So you provide patient material. So the point is that you have a live patient who one day may enter a trial. So he's fit enough to enter a trial. He has metastatic disease, for example. So you enter your patients. This material is sent to central labs, which can rotate. I mean, there's not a fixed thing. So if you have a really cool facility, a really cool idea, you can get part of the material and run your tests on it. The return to the clinician then who entered the patient is that he has all this molecular data comes back to him. So he knows now a lot of things about his patient that comes straight back to him. He can use that for his own approach potentially if he wants to try out something based on one of these features. He has this feedback of this omics on his patient coming straight to him. But on the other hand, the patient goes into a database of live patients, which we update every 6 months to know how they're doing. And that's where you can come in and mine for a trial. You say, okay, I want 10 live patients with this feature at that stage of the disease. So we do hope that those patients will actually get exposed to clinical trials. So that's kind of the win-win for the patient, win-win for the clinician, and us basically just facilitating this. Yeah. There's a website, there's folders, there's, yeah, if you go like EORTC SPECTA or SPECTA Color, there's the websites are there, all the contacts are there. In general, we're just setting up, or it's up and running, so I'm not going to say that we have been able to already prove exactly, which is what we want to do, is to prove certain mutations will predict certain drug responses. And that may not necessarily even be the case. We know there's a lot of problems there. And that's not really our aim either. Our aim, and I think where our deliverables and where we can be measured, at the end of the project, is to know where are we able to get good quality, molecular subtyping, which is state of the art, including unpublished, academic ideas like the ones we've discussed today at WIN. Have them running on an efficient way on thousands of patients every year, and these patients then being used by the community, either the academic community for your own trial or the pharma community for one of their trials. That's actually our deliverable. Afterwards, does the drug work in the patient? That's a different question. Yeah. But it's really getting more exposure of these patients to the drugs. Yeah. We get the patients on, let's say, early on in their disease, because actually we want to be able to offer them a lot of options while they're alive. And that's also what we tell them. We are going to be able to provide you with some profiling which is beyond what we could be doing ourselves in our hospital or whatever. And there's really only clear advantages to them. What we do make sure is that, of course, this material is always treated in an anonymous fashion, and the molecular data can't be linked to any of their features. Or we also have a process how to handle germline alterations should you find them. So the usual things you have to take care of. But basically, it's completely in the interest of the patient to get to know more about his tumor, and they're quite excited about it, actually. They ask for it. So. Yeah, maybe if I may interrupt, but it's also important. There's more and more, patients are aware of this through the media, and I'm not talking about the SPECTA, but just in general about molecular profiling. So they would tend to go out on their own, maybe, and even pay like outside companies. And we get that. We get patients coming with their full reports, and first of all, the report you can't interpret, because in the SPECTA, at least we interpret what we measure. We interpret. And the second thing is, imagine if a patient were to do that and send his material to 2 or 3 sources, he would not have any material anymore. So what we try to do is centralize whatever you do on this material is in one repository. There's one pathologist that handles the material of that patient, and we make sure it's efficiently handled. And I think that is also in the interest of the patient. Money. Because, as I described it, it's a facilitating platform. But if there's no end user or no clear benefit for an end user, there's no money coming in. So at the moment, we're fantastically lucky to have charity money, and specifically for the colon, this is Alliance Boots is sponsoring us. So it's really they do all kind of charity events all over the world just for the SPECTA Color platform. And for each disease, we have different charities. So at the moment, it's all charity money. Yeah, the viability of this all will be the pickup by society, maybe by even payers, regulators, pharma, whoever. When they get to see the advantage for themselves, then then it can be something that can be funded. But it's something you want to set up and keep on running forever. Actually, so that's the challenge. Again, for me, the deliverables are to have a live pool of patients that can get queried, and where actually all the parties are happy. For example, if a pharma wanted 20 live patients in centers with which they used to work with, maybe a phase 1 trial, difficult trial, that we can provide those. Yeah. But most, for me also, what the big challenge is that the omics data that we do is of good quality. So that's a big thing, quality assurance. So we have in this platform a lot of quality assurance going on. We talk to the regulators. We talk to we have the European Society of Pathology is integrated here. So we make sure that the quality has to be really high. And if we can find a way to deliver, harmonious quality over time for many complex markers, and have this open to new people and new ideas that want to come in with a new marker, and still we can keep on providing, a well-oiled machine, that would be the deliverable for me. If we can manage that, then I think we have what we wanted.